Welcome, everyone. It's a pleasure to have with us today Dr. María Blasco. She's the director of the Spanish National Cancer Research Center, CNIO, in Madrid. Just to summarize a little bit her trajectory, she's a natural from Alacant in Valencia in Spain, and she did her PhD in Madrid in the Centro de Biología Molecular with Professor Margarita Salas. From there, she moved to do a postdoc at Cold Spring Harbor with Nobel laureate Carol Grader. And from there, she moved back to Spain, in this case to the Centro Nacional de Biotecnología in Madrid. And from there, he moved to her position right now at the Spanish National Cancer Center, where she became the director. Maria, she has been a force in the field of telomeres. She's a molecular cell biologist, and she has been a force in understanding the function of telomeres and telomerases in biology, but also in cancer. And in cancer, she was in cancer and aging, and she was really a critical element to show that how short telomeres are sufficiently enough to induce aging, and how this can be reverted by the activation of telomerases, among many other things. In addition to her efforts in science, she has been also a force in two areas. One is supporting equality in science, and the second in the interface between society and science. And these are also important avenues right now for the world. I would say for the world in this time of crisis even more. She has received many recognitions for her discoveries, among these EMBO Gold Medal, the Josef Steiner Award, the Corben European Science Award, the National Research Award, the Jaume Premier Award, etc., etc. So I would not like to delay more her presentation, because we're here to hear about her science, and it's a pleasure to have her here. The title of her talk is going to be Targeting Telomeres in Cancer. Thank you very much, Maria.
Thank you very much, Manel, for the very kind introduction. For me, it's a pleasure to give this seminar at your institute. I wish I was there, but this is also fine. So I will talk about the targeting telomeres in cancer, but I will start with another story on aging that Manel already mentioned, that my group is also interested in aging, because it has led to the generation of a new co, and I think it's very exciting to be able to apply the research we are doing into really treatments. So if you see my screen, just to tell you that we think that biological aging is the cause of many diseases, and this also includes cancer. And therefore, it's very important to understand which are the molecular pathways that lead to aging, because this is at the origin of all these diseases. And in a recent, well, not so recent review, we went over all the molecular pathways that have been involved in aging. And one of them is the shortening of telomeres, that Manel already mentioned. We think that this is a primary cause of aging, because when telomeres are short, this leads to other molecular causes of aging that include genomic instability, stem cell exhaustion, cellular senescence, etc. So I think there is a lot of evidence that short telomeres really at the origin are one of the molecular origins of aging. There is an enzyme that is called telomerase. It was discovered in 1985 by Carol Grader and Liz Blackburn, and that's why they received the Nobel Prize for Medicine and Physiology. And this enzyme is able really to elongate telomeres when we are embryos, and in a way reset telomere length in each new individual. When we are born, however, telomeres start shortening, and we think that this shortening of telomeres, as I mentioned, is one of the causes of aging. When telomeres are critically short, this leads to disease. In the case of cancer, cancer is also associated with aging, but cancer cells, in order to be able to divide, they have to reactivate telomerase. And really, activation and mutation of telomerase gene in cancer is one of the most frequent alterations in cancer. So I will go back to that later. What I wanted to mention is that telomere length is very limiting for aging, because if you have a mutation in telomerase, and this happens in the so-called telomere syndromes, these individuals are going to have a premature loss of the ability to regenerate tissues and are going to have a number of diseases, including, for instance, pulmonary and liver fibrosis. Also, individuals that are not mutant for telomerase, there are many studies showing that shorter telomeres than normal are linked to a higher susceptibility to different age-related diseases, and telomere length is considered a biomarker of this molecular aging process. So we have shown actually that short telomeres are at the origin of aging by generating a telomerase deficient mouse model. This is already very old work, but it really demonstrated that short telomeres can cause aging. And also that telomerase is important for cancer, because if you have mice without telomerase, these mice are resistant to cancer. So telomerase or telomeres are a good target for cancer. So both in humans and in mice, telomere shortening causes aging. We last year also showed that not only human and mice, but in many different species, we found that the rate of telomere shortening was really predictive of the lifespan. And you can have, for instance, two species like the flamingos and the elephants, which are very distant in evolution, but they have the same rate of telomere shortening, and they also have the same longevity. So the rate of telomere shortening seems to be really a predictor of longevity in different species, highlighting again the importance of telomere shortening also in evolution to set the longevity of different species. What is interesting, of course, is that if you have telomerase that can elongate telomeres, is to see whether telomerase can be used to delay aging, and in this way delay age-related diseases. And we showed this for the first time in 2008. We were able to generate transgenic mice that were able to maintain the telomeres for longer times, and these mice also show an increased health span and a delay aging. You cannot make transgenic humans, but we, together with the group of Fatima Bosch in the Universidad Autónoma de Barcelona, we generated a telomere gene therapy strategy using adeno-associated viruses, and we tested whether treatment with this gene therapy was also able to delay aging in mice. We did the experiment in 2012, and basically we treated middle-aged or all mice with a single treatment with this telomere gene therapy, and we were able to delay aging and delay many diseases associated with aging, including cancer. So cancer was also delayed. This means that cancer is a disease associated also to presence of short telomeres and aging. Actually, these mice didn't have any bad secondary effect. The only thing that we observed is that these mice live longer. The 1-year-old group lived 24% longer, and even the very old mice, the 2-year-old group, lived 13% longer. What we have been doing since then is to try to use this telomere gene therapy for the treatment of different age-related diseases. We show that it was effective in protection against heart infarct in mouse models, also in aplastic anemia associated to very short telomeres. And more recently, we have been focused in pulmonary fibrosis. Pulmonary fibrosis is one of the most prevalent telomere syndromes. It was described that telomerase mutation lead in familial cases to pulmonary fibrosis. And this is for us very interesting to see how telomeres are at the origin of this pulmonary fibrosis, because even in the sporadic cases of pulmonary fibrosis, patients that have pulmonary fibrosis have shorter telomeres than the normal patients. So this disease is very prevalent and is increasing due to the demographic aging. And we wanted to demonstrate whether telomerase gene therapy could have a therapeutic opportunity here. So this is our view. Short telomeres may synergize with other agents that damage the lungs in inducing damage in the regenerative cell types in the lung, such as the alveolar epithelial cells, which will enter senescence, DNA damage, and because when telomeres are short, this impairs the regeneration of these cells. If you don't have regeneration, you're going to have a immune response and fibrillar recruitment and fibrosis. So the treatments that we have right now in the clinic are treatments that are not curing patients. They are somehow blocking the fibrosis, but because fibrosis we think is downstream of the real origin of the disease, which is associated to presence of short telomeres in the lungs, we think that this is why you have an impaired stem regeneration and you are not able to cure the patients with these treatments. So there is really a need of new treatments. So in order to be able to study this, we generated mouse models in which we induce fibrosis owing to short telomeres. So we use a dose of a damaging agent to the lung that is called bleomycin, which doesn't cause pulmonary fibrosis in control mice. So the control mice with this bleomycin do not develop fibrosis. However, when we put this in the context of the telomerase knockout, these mice develop fibrosis and they die of fibrosis, very similar to the human fibrosis with activated myofibroblasts, inflammation, and collagen deposits. So we have a sort of humanized mouse model that is developing fibrosis associated to short telomeres. And we now are going to treat these mice with the telomerase gene therapy. So we're using adeno-associated viruses, and we were lucky because the cell type that is mainly transduced by this adeno-associated virus are the alveolar type 2 cells, which are the ones that we know are important for the disease. So when we transduce with GFP, 80% of the transduced cells are alveolar type 2 cells. So we then put the telomerase gene, and basically what we do is that we induce fibrosis. We diagnose fibrosis by CT. We take only the mice that have the positive diagnosis for fibrosis, and we treat these mice either with the telomerase or with the empty vector. And already by following CT images, we saw that the ones treated with telomerase, they can completely revert the lesion. However, the ones treated with the empty vector, they retain a lesion in the lungs, and we see that the telomerase is somehow making this fibrosis to disappear. This at the level of CT, but also by histopathology. You can see that the mice that we treated with the empty vector, 100% of them have severe fibrosis, but the ones that we treated with TERT, half of them are completely cured, and the other half have only small patches of fibrosis. I would like to point, but I don't know how to point here, so sorry. So when we look at this at the molecular level, we see that telomerase is very fast. In 3 weeks is rescuing DNA damage, senescence, apoptosis in the lungs. We do a panel of pro-inflammatory cytokines. We see that this inflammation is being rescued by the treatment with telomerase. And even if we isolate the alveolar type 2 cells of the mice treated with telomerase, we see that there is a decrease in the DNA damage response pathways, immune signaling pathways. And we can also see how there is a rescue in all the profibrotic pathways when we treat with telomerase. So summarizing, this tells us that short telomeres are the origin of this pulmonary fibrosis. And if we correct that, we can correct everything else. So this means that everything else is downstream, and that's why we are very interested in applying this to humans. So the first thing we wanted to do is whether this could be dangerous. I mean, if you activate telomerase in the lungs, whether you can have more cancer. So and for that we did the following experiment. I can skip this. We did this experiment. So at the same time that we put telomerase, we activate KRAS in the lungs. So when you activate KRAS in the lungs, this is the Mariano Barbacid mouse model, you have lung carcinomas. And what we did is to have 2 groups, one in which we treated with telomerase before the oncogene, and the other one we treated with telomerase at the same time of the oncogene induction, to see whether telomerase was increasing cancer in this context. And what we saw is that the telomerase expression, either in the pre-treatment or simultaneous treatment groups, was not accelerating tumors at all. So it didn't matter whether you put telomerase or not in this KRAS mouse model that is forced to develop lung cancer. And also when we look at the tumors by histopathology, we saw that the telomerase treated did not have more carcinomas or did not have more adenoma. So it seems that this telomerase treatment, at least by this criteria, is not accelerating tumors in lungs, even if they have a KRAS oncogene activated. So for all these reasons, we funded a company which is called Telomere Therapeutics, and so we will start working very soon. So we hope to bring this to humans as soon as possible. And now we will move to the main subject of my talk, that is the role of telomeres and telomerase in cancer. I think you're all familiar. I already mentioned it, that telomerase mutations are one of the most frequent mutations in cancer. And this is because maintaining telomeres in cancer is going to allow for indefinite replicative potential of cancer cells. And this is mainly done through mutating telomerase, but it's not the only way. Actually, at the CNIO and in my group, in collaboration with the group first of Lopez Otin and Elias Campo, we found the first mutations also in one of the proteins of the telomere protective complex that is called sheltering. So sheltering is bound to the telomeric repeats and is actually exerting telomere protection. So one of these proteins is called POD1, protection of telomere 1, is also mutated in cancer. And first we found it in chronic lymphocytic leukemia, but then it has been found in many other tumor types. So at the CNIO, we collaborated in the CLL and the Li-Fraumeni-like syndrome that also has mutations in POD1. What is doing this POD1 mutation is acting like a dominant negative. In one hand, it generated chromosomal instability because this protein is very important to protect the telomeres, and at the same time is allowing for abnormally long telomeres, probably because it's allowing for a higher extension by telomerase or by recombination. So this is another proof that cancer cells need to maintain the telomeres, and they can either mutate telomerase or they can mutate these telomere binding proteins. And I will come back to this later with some unpublished data. So then I already mentioned that telomerase inhibition has been important in the field, but the truth is that one of the most advanced telomerase inhibitors basically has failed in solid tumors. It has only been effective in some myeloid malignancies, and we think that this is because if you inhibit telomerase, the killing of cancer cells will depend on the telomere length you have in the different tumor populations. So you will have first a lag period and the telomeres shorten, and because of the tumor heterogeneity, maybe you will be killing the cells with the short telomeres, but you would actually maybe select for clones with longer telomeres, and this may be actually dangerous because we know that longer telomeres could be a property of stem cells and maybe also of cancer stem cells. And also, if you inhibit telomerase, we know that there are alternative mechanisms to maintain telomeres that are based on recombination. So we think that maybe these things could have made fail the telomerase inhibitors in the clinic. So that's why we move to a different subject. Instead of inhibiting telomerase, we try to really destroy the telomeres of a cancer cell and see whether this has a therapeutic potential, this has a therapeutic window, and whether it could be effective in slowing or inhibiting the growth of tumors. We know in my group that removing any of the shelterings, because we have done knockouts for all of them, except for RAP1, which is not essential, removing any of the other proteins is killing telomere protection and is stopping the cells, making them enter into senescence or dying by apoptosis. So it's a very effective way to stop the growth of cells. We decided to focus in TRF1, which is binding throughout all the telomeres. Is the telomere repeat factor 1. And when you delete TRF1 genetically, for instance, you induce DNA damage in every chromosome end. So every chromosome end is like a double strand break, and this stops the cells very rapidly. And we have shown that this is, for instance, effective in mouse models of lung cancer, the ones I mentioned from Mariano Barbacid. And also we know that in my group, the TRF1 actually is a stem cell marker, is enriched in the stem cell population. And we thought that this made TRF1 a good target for cancer because it's present or is highly expressed in the more stem populations. And also inhibiting TRF1 is really effective in slowing down the growth of tumors. So we decided to test this in glioblastoma. This is already a published work because this is a very aggressive tumor and is known to have a lot of tumor heterogeneity, and it's known that there are a population of stem cells, the glioma stem cells, that are able to recapitulate the tumor even though you treat with chemotherapy, radiotherapy, surgery. The tumor always comes back, and this is thought to be due to this glioma stem cells. So we wanted to see whether deleting TRF1 could have a therapeutic benefit in mouse models of glioblastoma. And we show actually, we use mouse models of glioblastoma that are inducing the tumors in the brain by expressing oncogenes, DGFB and PDGFA. They are going to be expressing glial progenitors, and this makes the mice develop different times. Depending on the oncogene, this must develop glioblastoma. So we check whether TRF1 was overexpressed, and this would be in agreement with our hypothesis, and TRF1 is overexpressed in these glioblastomas, suggesting that is important for the growth of these tumors. And I'm going to go fast because this is published, but we saw that if you delete TRF1 at the same time that you induce the oncogene, either the A or PDGF-A or B, we did it in 2 different models. We have different tumor latency to see whether the effect was the same. And we could see a very clear increase in survival of the mice when we deleted TRF1 at the same time that we induced the oncogene. So this would be like, if you want, the preventive model. Actually, all the tumors that appear in the TRF1 deleted mice were escapers. There was not a single tumor that actually deleted TRF1 and could grow. So then we moved to the treatment model, in which we first induce the tumors, and once the tumors start to grow, we delete TRF1. And in this case, we also saw a very clear benefit. We saw that the deletion of TRF1 after the induction of the tumor and after the tumor was already present also increased very significantly the survival of these mice, so supporting that TRF1 could be a good target for glioblastoma. So we wanted to see whether this was acting at the level of the glioma stem cells. Well, first of all, the mechanism, I already explained it, but just for you to see it. So when you delete actually TRF1, what is happening actually? Of course, you are blocking proliferation, but what is really happening at the molecular level is that you're inducing DNA damage. You're inducing DNA damage in the tumors, but this DNA damage is specifically located at the telomere. So when we look at the colocalization of the damage with the telomeres, the so-called TIFs, telomere-induced DNA damage foci, you can see that the deletion of TRF1 is inducing a very specific type of damage, that is damage at the telomeres specifically. So this is different from a DNA damaging agent that is inducing damage all over the genome. When you delete TRF1, it's only inducing damage at the telomere, so it's more specific if you want. We saw that this actually was acting also at the level of the glioma stem cells. So we isolated glioma stem cells, and then we put them into the brains of mice, and they are going to develop glioblastoma. But if you delete after you put these cells in the brain, because they are conditional for the TRF1 locus, then we saw that these mice could survive. So this is telling you that it's acting at the level also of the glioma stem cells. Were there any safety issues? Well, in mice, we didn't see any safety issues of deleting TRF1 in the brain. We have done this experiment. We deleted TRF1 specifically in the brain of newborns, and then we led these mice to age, and we didn't see any defects in survival or in different cognitive tests. And we have even deleted TRF1. We published this more recently in the tumor-prone models, because we thought maybe if you delete TRF1 is going to generate genomic instability, TRF1 deletion, and maybe this could synergize with mutations in tumor suppressor genes like p53 or p19R, p16 in inducing more tumors. And for that, what we did is to induce the TRF1 deletion in the context of mice deficient for INK4A or for p53, and we didn't see any effects on the survival of these mice. So this was not accelerating tumors induced by p53 or p19 and p16. So actually, we saw some phenotypes in the mice. Of course, you are deleting TRF1. This is going to induce some damage, but the phenotypes were mainly in the skin. So we saw, in the case of the INK4A, we saw that the mice deleted for TRF1 had white hair. In the p53 background, we didn't see much phenotype, maybe some hair loss. And so this is telling you that, of course, chemotherapy, for instance, with TRF1 inhibitors may have some effects in the skin, etc. But we didn't see any major effects on survival in this experiment of other experiments. So this has this is just a summary that the genetic deletion of TRF1 in different mouse models is showing impairing the tumor growth, and this is because it's inducing DNA damage, decreasing stemness and proliferation. And then we also wanted to see whether we can find inhibitors, chemical molecules that will decrease TRF1 levels and could be used in patients. And at the CNIO, we have a drug discovery program, and they screen for molecules that were able to decrease TRF1 levels. So we have a screening platform in which we have TRF1 fluorescent, because we have a knock-in mouse for TRF1. Whenever TRF1 is expressed, you have the green fluorescent protein. And you can look for molecules that decrease the expression of the green fluorescent protein at telomeres. So this would be TRF1. And we hope that these molecules will be inducing TRF1, inhibiting TRF1, inducing DNA damage specific at telomeres, and will inhibit the cell growth by inducing senescence, apoptosis, and mitotic catastrophe. And we found two series of inhibitors, series 1 and series 2. Series 1, we have characterized it very well, and they are inhibitors of PI3K alpha. With this, we actually discovered something that we didn't know, is that TRF1 is regulated by AKT phosphorylation. So TRF1 has a number of phosphorylation sites for AKT, and that's why when you inhibit PI3K alpha, you inhibit AKT phosphorylation, and you are destabilizing TRF1 from the telomere, and that's why PI3K inhibitors are actually inhibiting also TRF1 at telomeres. We found 3 phosphorylation sites, and we demonstrated that TRF1 is phosphorylated at these sites by mutating these sites in vitro, and that when you mutate these sites at TRF1, these are in vitro experiments, you have a lower protein stability, you have less binding of TRF1 to the telomeres, and by expressing these mutant proteins in knockout cells, we also found that they were impairing the binding to the telomeres. This is in mouse. We have found now recently in humans, this is still unpublished, the paper is submitted, that also in humans, TRF1 is phosphorylated by AKT, and we have used CRISPR-Cas to make knock-ins in human cells in these phosphorylation sites of TRF1. And again, we find that these phosphorylation sites are important in vivo, because if you have a knock-in in these sites, you induce more DNA damage at telomeres. And we also found telomere length effects. So this phosphorylation of AKT by of TRF1 by AKT is important in vivo. If you generate knock-in cell lines, and we are generating now a knock-in mouse to be able to prove this further. So we also have a second series of inhibitors. We have a cache impulse to characterize the mechanism of action. I will not mention much now. These are not inhibiting anything known, so there is more interest there. And the same as I showed you with the genetic deletion, when we use the inhibitors, the chemical inhibitors, we see that this impairs the growth of glioma stem cells. And we have also done in vivo experiments with xenograft models in which you can see how this treatment with these chemical inhibitors that inhibit TRF1 protein are blocking the growth of the glioblastoma. What we have done afterwards is that we published this last year, was to screen for a collection of FDA-approved drugs to see how many things we can find that actually inhibit TRF1. And to our surprise, we found many different cancer pathways that are important for TRF1 inhibition, which is, I think, telling you of the importance of this protein or the importance of telomere protection for cancer. Because there are many known pathways, particularly we found the RAS pathway. Of course, we already have found the PI3K pathway by the RAS pathway. We found MEK and ERK and BRAF, even though it's not here. We are studying also TRF1 regulation by BRAF. And this you can see here in the right, the TRF1 protein and all the phosphorylation sites by different important cancer kinases, which is telling you the importance of telomere protection for cancer. So I wanted to finish with something that is already unpublished, and is that we are trying to generate a mouse model to understand the role of Pol1 mutations in cancer. Remember, I told you that cancer cells not only mutate telomerase, they can also mutate preferentially one of the telomere binding proteins that is called POD1, protection of telomerase 1. We have found this first in CLL, and then we also found it in Leukaemia-like syndrome, the one that is not associated to p53 mutations. So this means that being a carrier of a mutation in TRF1 is increasing the susceptibility to many cancer types, like in the case of the Leukaemia-like families. These families also even have a very rare tumor that is cardiac angiosarcoma. And we thought it would be interesting to try to make a knocking mouse carrying mutation in the POD1 gene that we found in the Leukaemia-like families to try their different therapies or to understand the origin of this disease. So these are the different tumor types where mutations in shelterings have been found, mainly in POD1, even though in melanoma they also have been found in 2 other shelterings, RAP1 and TPP1, but mainly POD1 is the most mutated protein in cancer. And we have decided to study one of the mutations, arginine 117 to cysteine in the protein in POD1. That's one of the most frequent in Leukaemia-like syndrome. Just to remind you what we found in the humans. So in humans, these mutations induce decreased POD1 levels in the carriers, and these carriers have longer telomeres when you look at telomere length in blood, and they have an increased telomere damage. So these are people that carry this mutation in POD1. And so we generated the knocking for POD1. In the mice, there are 2 POD1 proteins, more complicated than humans. The POD1A is embryonic lethal, and POD1B is viable. So we thought that we should make a knocking in POD1A, which probably is the protein equivalent to POD1 in humans. So this is the results in MERS. So again, when you have a knocking in the double the homozygous knocking is lethal. So we could only study the heterozygous mice. But the heterozygous mice, you can see that even though we don't see an increase in total damage, if we look at telomere damage, we can see that telomere damage is induced in these carriers, the heterozygous carrying this mutation, same as in the patients. Also, they have more telomeric aberrations, the same as in the patients. These are in the MEFs, in the mouse embryonic fibroblasts. But if we this is in a p53 wild type. If we do this in a p53 knockout background, we also find the same. We see more telomere-induced damage, and we see more telomeric aberrations. So this is telling you that this allele is generating chromosomal instability, the same as in the telomere damage, the same as in the humans. And we also found the presence of these very long telomeres. We call them super long telomeres, the same as in the patient. So you have some chromosomes with really huge telomeres, and you can see this by what we call the coefficient of skewness. We find telomeres which have very long telomeres. We have also looked in the mice, and in the mice again, when we look at the lymphocytes, we see more telomeric damage. So we see that this mutation, the mice that are heterozygous for this allele, they have more telomeric damage, and they also have longer telomeres in the blood, in the white blood cells, like in the patients. So we have also found increased tumors in these mice. This is still unpublished. We are characterizing that, but we see, I think the values are going to reach significance, but we see increased hemangiosarcomas. Remember, sarcomas are one of the tumors associated to this Leukaemia-like syndrome, but we also see a tendency to have more carcinomas, for instance, liver and lung carcinomas, which in mice is difficult to see. We see that these mice carrying the POD1 mutant allele in heterozygosity have more of these carcinomas. So I think this mouse model will be useful also to understand cardiac hemangiosarcomas, because we find in these mice many of these hemangiosarcomas in the heart, which is something that happens also in the patients, and we see also in the mice. So I think this is my last slide. I want to thank the people in my lab that has been participating in this work, all the gene therapy work. I would like to thank Paula Martinez especially, and now also Sergio Piñeiro. And the work that I showed you on the Polygon mutations is also Paula Martinez, the one that is leading this work in the lab. So thank you very much for your attention. This is my acknowledgement funding slide. So thank you for attention. I think I went very fast.
Oh, yes. Don't worry, it was great. Thank you very much. Thank you very much, Maria. So I would like to remember to all the attendees that I forgot this to mention before, that you can ask your questions using here the online system, and I will ask the questions when I have them here. So we have just a few now. And one first, I have a comment because we had recently Angela Nieto, and she talked about fibrosis in the kidney,
yeah.
and you have this model of fibrosis in the lung. So these are very similar. That was an EMT model.
Yeah, this is very interesting. Actually, I didn't show this here. This is right now submitted, but we have also shown that short telomeres are at the origin of kidney fibrosis. So we have now a kidney fibrosis model in which we apply a slow dose of the DNA of a damaging agent to the kidney, folic acid, which at this dose doesn't do anything to the wild type kidney, but to the kidneys with short telomeres, it induces fibrosis, full-blown kidney fibrosis. And actually, we have a study there whether it's inducing these short telomeres are associated to an epithelial to mesenchymal transition. And actually, this at the end is the subject of the paper. We see that short telomeres are inducing epithelial to mesenchymal transition in our kidney model. I didn't want to put too many things in the talk, but yes, kidney short telomeres are also inducing or synergizing with damaging agents to the kidney to induce kidney fibrosis. And we see that they are also at the origin of the epithelial to mesenchymal transition in the kidney model. We are looking now also in the lung model to see whether we also find an increased expression of all the EMT genes. But yeah, this is interesting. So somehow short telomeres, we could relate them also to the induction of this epithelial to mesenchymal transition. Even in the absence, we found these changes in expression of these genes, even in the absence of the damaging agents to the kidney. So even just with the wild type and the telomerase deficient mice, when we look at the kidneys, we see an overexpression of the epithelial to mesenchymal transition genes. So I think this is interesting and connects to what Angela Nieto is doing. And of course, I knew her work.
Okay, excellent, thank you. So you describe drugs that target elements of the machinery of the complex of the telomeres. What about the drugs that targets telomerase itself?
Yeah, I think I maybe went too fast, but I think it has been sort of a disappointment. The drugs that have been more advanced in treatment of tumors, the clinical trials have failed, so in solid tumors. So telomerase inhibitors have not worked in treatment of solid tumors, only myeloid malignancies. There was some benefit of treating with telomerase. And the reason for that, I think, is because there are three reasons. One is that when you inhibit telomerase, you need time until the telomeres shorten, and this time may be too much if you want to cure a patient. Two, that the tumors are heterogeneous, and when you treat with a telomerase inhibitor, you may be killing the cells with the short telomeres, but you may be selecting for clones with longer telomeres that could be even more dangerous than the clones with the short telomeres. And three is that when you inhibit telomerase, cells can recombine using homologous recombination. They can maintain telomeres also. So you may be selecting for mechanisms of homologous recombination between telomeres to maintain telomeres. So that's why we thought about, instead of targeting telomerase, targeting telomeres, because if you directly try to destroy telomeres and you show that this has a therapeutic window, and we have shown this in several mouse models. I discussed the glioblastoma models. So then it doesn't matter, I mean, whether you have tumor heterogeneity, because you're going to kill all cells that divide basically in the tumor when you inhibit sheltering. And also there is no chance for homologous recombination to appear to rescue this, because you are blocking the telomere protection. You basically don't care about which is the telomere length. You are just trying to destroy telomere protection in the tumors.
Okay. Thank you. Another question here says, do POD1 cancer have ALT-like features?
Yeah, we think it does, and we are addressing right now specifically with the knocking mouse model that we have generated. We can measure telomere recombination. There is a technique that is called co-fish that allows you to see the recombination between telomeres. We don't know yet the result, but yeah, the fact that we find these very, very large telomeres, so like huge telomeres in the chromosomes of these POD1 knocking mice, is suggesting that maybe POD1 mutation is also favoring recombination.
Okay. What about targeting Symmic TRF1 interaction?
Mm-hmm. Well, we have not thought about that, but it could be interesting. Yeah, we are more focused now in trying to target. Actually, what we are doing right now is trying to block TRF1 by sort of drugs that impair TRF1 dimerization or TRF1 binding to the telomere, because we thought that these drugs would be more specific. The thing that we have found with TRF1 with inhibitors of TRF1 is that we find many of the we find ERK inhibitors, we find MEK inhibitors, we have PI3K inhibitors, and I mean, this is fine if you want to combine combine drugs. But if you want to have a TRF1 only, a TRF1 specific inhibitor, we are going through to have more specific assays to find inhibitors. Like we want to block TRF1 dimerization of TRF1 binding to the DNA, and this is what we are doing now. This is still ongoing, so we don't have yet something that I can tell you. But really, we want to go for TRF1 only inhibitors. Because we think that these will be the ones with more value right now. Then you could combine them with other inhibitors, but I think what has probably commercial value right now is to have a TRF1 only inhibitor.
Yeah, I agree. Yeah. Here another question says from an attendee, TRF1 is necessary for maintenance stem pluripotency. Yes. Can you exclude a non-telomeric effect of TRF1 in the inhibition of GBM?
TRF1 is only present at the telomeres. I mean, when we do chromatin IP and we find where is binding, I mean, TRF1 throughout the genome, TRF1 is binding to the telomeres. It's not binding elsewhere. So could be, I mean, we cannot rule out there could be any effects of TRF1. Yeah, for instance, related to the role of TRF1 in pluripotency or stem nets. But yeah, no, I think it's right. I mean, we think that TRF1 inhibition is inducing telomeric damage, but it also could be affecting the role of TRF1 in pluripotency, which we have shown that actually, I didn't have time to mention this, but we published this in eLife last year, that when you remove TRF1 from the telomeres, there is also an effect on the expression of the Polygon proteins, and actually has sort of a butterfly effect, affecting the expression of genes important for pluripotency. So yeah, I think yeah, you're right, and I think that probably the effects on glioblastoma they are related to induction of telomere damage, but it could also be related to this role of TRF1 in modulating the levels of Polygon proteins that we have found that this is sort of mediated by the terras. The terras are long non-coded RNAs that are produced from the telomeres, and when we delete TRF1, we see an increase in terras, and these terras have been shown by us and others to bind throughout the chromosome arms, and in particular regulate Polygon.
Because the questions are now coming in an exponential manner here in the chat, so yes, I'm going to select 3 or 4, and because I want to tire the speaker.
Yeah.
So one is, what is the cause of shortening of telomeres in those sporadic cases of pulmonary fibrosis?
Yeah, well, it's a good question. I think pulmonary fibrosis is a disease associated with aging, so the risk of developing pulmonary fibrosis increasing as you get old. So telomeres shorten associated with aging, and I can imagine that if you have been exposed to damaging agents to the lung, I don't know, smoking or pollution or asbestos, this could be killing your lung cells and inducing a higher turnover of your lung cells throughout your life because you have been exposed to smoking or pollution or asbestos, for instance. And this higher turnover is going to increase the rate of shortening in the lung. So this could be a cause why people with sporadic pulmonary fibrosis could also have short telomeres because of a higher exposure to damaging agents to the lung.
Another question that appears many times, this type of question says, if you inhibit TRF1, then are also increasing the aging of the animal?
This is a very good question. I show you the survival curves for mice wild type or deleted for TRF1 in the adulthood, let's say. I mean, if you delete TRF1 at with a regular conventional knockout, it's lethal. The mice don't go through blastocysts, so it's completely lethal. But if you delete TRF1, and we have done this in an ubiquitous manner in mice, we don't see much effects on mouse survival. We see some. This, of course, had to be shown for the therapeutic effects, and is published in the Cancer Cell, and I think it's also published in the previous paper in Embol Molecular Medicine. So we don't see effects in the survival, but we see some phenotypes. So we see some phenotypes in the skin, in the bone marrow, some aplasia, but these phenotypes are not prematurely killing the mice. Even I already showed you that we published last year that also if we delete TRF1 in tumor-prone models, that we thought maybe this is going to increase cancer in a 53 background, we didn't see more cancer. So I think there is really a therapeutic window for inhibiting TRF1 for tumor treatment.
Yeah. What about targeting TRF2? Similar spectra or phenotype?
We are doing right now, actually, we have a project for comparatively deleting all the shelterings and seeing which one is best. I would say TRF2 is going to be very similar to TRF1. There is a difference. TRF2 doesn't have the role that TRF1 has on stem cells and pluripotency. So maybe that's why we chose TRF1 over the others. That TRF1 is a target of OCT4, is essential for pluripotency. So it seems to have this extra value of being important for stem cells and for pluripotent cells, which other shelterings don't seem to be important. So that's why we started with TRF1, but we want to see also the value of inhibiting the other shelterings. And we are doing this, generating, actually, in the glioblastoma model, deleting the different shelterings. It's a boring experiment, but
it's a good question because you're doing an experiment. It means it's a good question. It's clear.
Yeah.
So then I will take the privilege to ask the last question for a Friday. So what about beyond cancer? So, for example, dementia. What is known about Alzheimer and telomerase? There isn't a role there because this is a big human health issue right now.
Yeah, I think so. I think so. I think the situation is a bit confusing because there are many papers showing correlations with short telomeres. Others don't confirm the correlation with short telomeres. But at least in we published a paper, I think it was a couple of years ago, showing the different cognitive phenotypes of the telomerase deficient mouse model. So even in the telomerase deficient mouse model, there are a number of cognitive defects, which is suggesting that the short telomeres could be also the origin of cognitive defects in mice or Alzheimer's, for instance. Actually, this is something that we are not actively pursuing right now, but I think is interesting to understand. I mean, for instance, which cell type in the brain could be more relevant for Alzheimer's? Is the microglia, are the neurons? So it would be interesting to induce telomere shortening in different brain populations and see, which ones are more relevant. I think this is something that is of interest.
Thank you very much, Maria. I think we have learned a lot about many things, about the cell biology.
I went too fast, but we had more time for questions. So.
Yeah, but I think that was to the point, and also is also relevant in the provided time. So I would like to thank you again for your talk, and please congratulate the speaker. Thank you very much.
Thank you very much to all of you. Have a good weekend.
Until next time. Bye.
Bye.